All right, welcome everybody. Thank you for braving the cold and making it out here. And there's some nice warm coffee and tea in the back to warm your hands for grand rounds. So very delighted today to have a very special speaker, Dr. Bob Dickson, who will be talking to us, and introducing Bob is Dr. Megan Trainor, not the singer. Yeah. Yes. So Dr. Trainor in Hem-Onc and palliative care.
Thank you, everybody. So I'm here to introduce Dr. Bob Dickson, who I met at the University of Michigan. He is newly, I believe, the recently endowed Galen B. Tow's Legacy Professor in Pulmonary and Critical Care at the University of Michigan. He's an internationally recognized leader in pulmonary and critical care medicine with a pioneering focus on the microbiome's role in lung disease and critical illness. His research spans molecular microbiota characterization, animal modeling of sepsis and lung injury, human clinical trials, over 125, I counted at least, peer-reviewed publications cited well over 12,000 times. Dr. Dickson's work has significantly advanced our understanding of diseases like ARDS, sepsis, pneumonia, lung transplant rejection, and his research is supported by the NIH, the European Commission, institutional funding, really underscoring its global impact. And in addition to his research, Dr. Dickson is a dedicated educator and mentor. He's the Associate Program Director of Pulmonary and Critical Care Fellowship Program at Michigan and the Director of the Multidisciplinary Training Program in Lung Disease at Michigan. He also holds leadership roles as the Associate Chief of Research for his division and Deputy Director of the Weil Institute for Critical Care Research and Innovation, as well as, I think, a project of love, you are the county TB doc in Washtenaw County. So we are so fortunate to have him come and talk to us and learn from his expertise. So please join me in welcoming Dr. Bob Dickson.
Thank you very much. Audio check, you can hear me okay? Great, so thank you very much, Megan, Dr. Trainor, and Lynn, Dr. Schnapp. It's lovely to be here. I feel like in a lot of ways, University of Michigan and University of Wisconsin are Big Ten sister institutions, and we have a lot in common, both intellectually and in terms of temperature as well. So it's a delight to be here and talk about some work. As you just saw, I have no financial disclosures. I do have an important disclosure. I am a lung doctor, and I'm going to be talking about multi-organ dysfunction, end organ failure. In my heart, in my head, like Lynn, I'm thinking about the lungs. It's the most important end organ. But you can substitute for a lot of what I'm saying, your kidney, your heart, your brain. There's other important end organs as well. I also have to disclose, the title of the talk is The Microbiome's Role in Critical Illness, by which many of you are probably assume I'm talking about the gut microbiome. And for the purposes of this hour, you're right. I'm talking about the 2 to 3 pounds of bacteria in your lower GI tract and what they have to do with organ failure in the ICU. But I actually got my start in research studying the respiratory microbiome. I was taught in med school that the lungs are sterile, and we spent the last 15 years debunking that and showing that even in healthy volunteers, we do find a signature of bacterial DNA reflecting bacteria that are actually alive, viable, metabolically active, immunologically consequential, variable across patients, predictive of clinical outcomes, and maybe involved in disease pathogenesis. And I think it is super exciting and would love to talk to you about it for an hour, but there just isn't enough time to cram it all into 1 hour. So have you back in a few years, maybe in the spring or summer when it's warmer out, and I'll talk to you all about that. But today my focus is on gut microbiota and what they have to do with why organs fail in the ICU. And I'll do that in 4 parts. I'm first going to convince you that despite what I put in my grants, this actually isn't a brand new idea that is highly innovative. This is actually a very old idea from the 1800s that has been rigorously studied and provocatively might be the most thoroughly studied phenomenon in critical care medicine. I'll explain that. I'll try to convince you that the clearest way to think about the gut microbiome is as an organ that has a function and a dysfunction that we should interrogate and maybe even think about targeting therapeutically. I'm going to argue it's the hidden driver of heterogeneity, which is the most exciting and interesting topic in critical care medicine research now. And finally, I'll argue that the gut microbiome is a therapeutic target, that not just something we should target over the horizon, but something we're actually already targeting now blindly with important consequences. So starting from the top, arguing that the gut microbiome's role in critical illness is an old familiar friend. So if you go back to the 1890s, Eli Metchnikoff, who the immunology nerds in the audience will know is sort of the father and founder of innate immunity, discovered the macrophage, he was a student of Pasteur. He said, hey, you've got a lot of bacteria in your lower GI tract, and we're not all infected all the time. So the immune system must be doing a lot of work keeping that from happening. But boy, if you get stopped up, and if you have translocation, if you have leakage of all those bacteria and their bacterial products throughout the body, that probably is not a good thing. You probably get systemically inflamed. And this is before we had a sort of modern conception of sepsis, but really laid out what I think in retrospect is probably right on. In the 1950s, Jacob Fine, I'll show a little more of this data, and his group in Boston, were studying not an infectious model, but traumatic shock. He was interested in the idea of the golden hour of resuscitation. So if you're in trauma and you lose a lot of blood, if they can stop the bleeding and give the blood back, typically people do okay. But if you're hypotensive for a while, it's hard to resuscitate a patient. Even if you restore their blood volume and stop the bleeding, they'll still develop progressive multi-organ failure. They wanted to understand that. And what he found was, in their animal models, if you pre-treated the gut with antibiotics, the animals did better. You could turn a lethal model into a non-lethal model. But it had to be beforehand and it had to be enterically. It had to be given into the gut, suggesting it really was something about those gut bacteria playing a role even in a sterile model of infection. And that's been an incredibly robust finding. So across shock models, across species, across ways of modulating the microbiome, it's really been borne out. That created a lot of energy in the 70s and 80s. And John Marshall, famous critical care investigator from Canada, said, the gut is the motor of multi-organ failure. And it gave rise to this, and this is, I find it fascinating. I'd love to spend an hour. I don't know if anyone in the room studies implementation science, but I think that this is the most fascinating example of it. Quick show of hands. How many of you routinely use selective decontamination of the digestive tract in your clinical practice? Of course, none of you. How many of you know what it is? Relatively, yeah, just a handful. And yet, this is, I'm going to argue, the most thoroughly studied intervention in critical care medicine. So back in the 80s, early 80s, late 70s, the Dutch are major advocates of this, inspired by this work and the idea that leak of bacteria from the gut to end organs might cause secondary infections and organ failure. Basically did to humans what Jacob Fine was doing to his dogs in his animal models. When they would come into the ICU, they would pre-treat, they would treat the gut with non-absorbable antibiotics. They were selective, they were protecting the anaerobes and suppressing the gram-negatives, right? So the proteobacteria, the inflammatory gram-negative rods. By selectively suppressing those, they found that they could prevent secondary infections, so dramatic drop in VAP and bloodstream infections, and also a drop in mortality. And the rest of the world just doesn't believe it. And the Dutch have studied it over and over and over. And here's a meta-analysis from JAMA, not an obscure journal, showing that, and I'm not kidding, it's over 30 RCTs, I think 25,000 patients, 99.3% posterior probability it's associated with lower hospital mortality. It's on the order of 2 to 3% mortality advantage to this. And no one in the world does it outside of the Netherlands. It's this amazing implementation science thing where we have all these practices that we take up and implement with obviously much weaker data for this. And then just because of what you might imagine, people have a perception that you're throwing antibiotics at patients. We're supposed to be stewards. You're going to be promoting resistance. Rigorously studied, no increased antimicrobial resistance. There's actually fewer cumulative antibiotic days in the SDD arm in these patients because there's so many fewer secondary infections. And yet, it's just not taken up worldwide. So I guess I am advocating for us considering SDD, but that's not the point of my talk. My point is, this is really not a new idea, and there's actually decades of experimental and human evidence to suggest there might be something behind it. So not a brand new idea, but if any of you are reviewing my grants, please still give me a good score for innovation. Still groundbreaking. All right, the gut microbiome as an organ with multiple functions. So if you ask me a simple, straightforward question like, what's the mechanism? How does the gut microbiome contribute to organ failure in the ICU? I'm not going to give you a simple linear answer. To me, that would be like saying, why does kidney failure matter in the ICU? Well, the kidney does a lot. I'm sure there's nephrologists in the rooms. It's not just regulating volume status. It's not just regulating pH. It's not just regulating electrolyte homeostasis. It's not just making erythropoietin. It's doing all these things, and when you lose it, a lot of things go wrong. I think of the gut microbiome that way. It's not a single bug. It's not a single metabolite. It's not a single immune pathway. The way that we all know intuitively is colonization resistance against potential pathogens. So this is the one that when you give your patient antibiotics in clinic or in the unit, you know you've just increased their risk of C. diff, right? And it's intuitively, all of those protective, non-pathogenic anaerobes are taking up space, and when you deplete them, opportunistic pathogens like C. diff can bloom and take over, right? That's old news, nothing controversial about it. Should have been enough to make us much more responsible about stewardship in the ICU, but that's not the end of the story. direct translocation of gut bacteria. So I mentioned this is what inspired the initial SDD, selective decontamination of the digestive tract studies, what Jacob Fine was thinking was going on. And it's got some meat on its bones. So again, since the 50s, we've been asking why modulating gut microbiota changes end organ failure or lung injury. So I mentioned this Jacob Fine story where he would take an experimental model of hemorrhagic shock. The dogs get hypotensive for an hour and then get resuscitated. A lethal model, 90% lethality, that if you pre-treated the gut with antibiotics, you could turn into a non-lethal model. And as I mentioned, a very robust, consistent finding across organ systems, across species, across end organs. Of course, for Lynn's sake, I'm showing lung injury here, but it's true for kidney failure, encephalopathy, mortality as well. Intraperitoneal E. coli, ischemia reperfusion, bradykinin, endotoxin. Basically, any way you can make an animal systemically ill, if you dampen down the bacteria, especially the inflammatory gram-negatives in the gut, they tend to do better. And the thought was it's because of translocation of the gut bacteria. There's not much linear distance between the gut lumen, where all those bugs are, and your systemic circulation and your lymphatics. So if something compromises that, and we know in the ICU that mucus layer sloughs off. We're not feeding them, that's part of it, but literally that protective mucus layer goes away and the wall gets thinner. And you can actually measure permeability. So this is very old data, well, relatively old data from the 90s. Depends on if you were conscious in the 90s. Quantifying permeability of the gut. So you give people non-absorbable sugars in their gut, and you can measure it in the blood and the urine and see how much of it was absorbed. The sicker you are, so the worse your multi-organ dysfunction, the leakier your gut. This is very well recapitulated in animal models. But when they systematically studied this, they didn't find it. So these are looking at patients at high risk for multi-organ failure sepsis and collecting portal vein cultures and systemic peripheral blood cultures. No evidence of translocation of gut bacteria correlating and predicting. But as you might guess, based on who's giving the talk, this was limited by their dependence on culture-based techniques, which are great for a lot of applications. It's still our go-to assay for a lot of aerobic pathogens, but not a great assay if you want anaerobic pathogens from the gut. That's just really not what our clinical culture protocols are up to. So some recent studies by our groups and others have, I think, rekindled the idea that gut translocation is actually an important process in sepsis and multi-organ failure. In 2016, one of our papers looked at this in both animal models and humans with ARDS. So in whatever model of sepsis we use in mice, if you look at the lung communities, so we're using homogenized lung tissue and just characterizing the lung bacteria using 16S sequencing, the workhorse assay for microbiome studies, what we find is that the lung communities are distinct from all of the appropriate control groups. It's not the antibiotics we co-administer, it really is the sepsis itself. And it's not a random difference, it's really gut bacteria that are enriching the lung. So to give you an example, this Bacteroidetes taxonomic group, so you can see it's about 15 to 25% of all the bacterial reads in the lower guts of these animals. When we look in the lungs of these animals, it's not there at baseline, it surges during sepsis, and it's gone by week 2. And because we can sample the upper respiratory tract, we know it's not coming from the top down, it's not aspiration in these mice. So it really does seem to be bottom up when we see translocation of gut to lung. And in humans, we had BAL specimens from over 100 patients with ARDS. We find a closely related taxonomic group, this Bacteroides, that's not a background contaminant, it's not present in healthy subjects, and about 1/3 of patients with ARDS had the same bacterial group in the lung. And it correlated with how systemically shocky you were. Your serum TNF correlated with how much of this gut bacteria we found in the lung. So all consistent with the hypothesis that gut leak, so translocation of gut bacteria, some of it, most of it probably non-viable, but some of it viable in bacterial products is translocating to lung and maybe contributing to lung injury. A couple years later, so Carolyn Calfee and her group at UCSF looked at another population that's at high risk for severe lung injury in ARDS trauma patients. She looked at tracheal aspirates and asked, do the identity of the bacteria in the lungs predict who does and doesn't develop ARDS, so severe lung injury? And the answer was the bacteria do, and it was specifically Enterobacteriaceae. So I know I can put you to sleep by making you memorize bacterial names, but you've heard of Enterobacteriaceae, important, common, gram-negative, lower gut bacterium. That was the most discriminating taxonomic group in terms of saying who would and would not subsequently develop ARDS. Then in partnership with Louis Bos from the Netherlands, we looked at mini BAL specimens from mechanically ventilated patients. We excluded patients with any clinical suspicion for pneumonia. So what I'm showing you is not just a very expensive, long-winded way of saying this patient has pneumonia. What we found was that the lung communities were different in the patients who did and didn't develop ARDS. And remarkably, it was the exact same taxonomic group, genetically identical to what Carolyn and her group found in her population. So again, sounds kind of obvious when you say it this way, but having lower gut bacteria dominating the respiratory communities in your critically ill patients is not a good thing. And lo and behold, it does predict ARDS. But to remind you, these patients didn't have pneumonia by any clinical appreciation, right? Like by culture, by X-ray and inspection. And it had prognostic implications. So all of these patients, if we quantified how much bacterial DNA was in the lung, the more you had, the worse you did. So this is in terms of whether you're alive and off the ventilator at 28 days. So again, having gut bacteria and having a lot of it in the lungs is a bad thing, correlates with severe lung injury, and is a sign that you're not gonna get off the ventilator. Here's some unpublished data. I'm showing you the end organ. That was just a lot of lung microbiome data. And you can hypothesize it came from the gut, but can we catch it in transit? Can you actually measure it in the blood of patients at risk? So this is unpublished data. We looked at blood, peripheral blood taken from a bunch of patients in our emergency department and University of Mississippi. Many of these patients had sepsis, but not all did. And if you line them up by their SOFA score, so for the non-critical care docs, this is basically a rough and ready measure of organ failure. How sick are you? 0 is hopefully everyone in this room, and 15 is essentially you have no remaining organs functioning, right? The sicker you are, the higher the serum bacterial DNA diversity. So this is true both for bacterial burden, how much DNA we found in the blood, bacterial DNA we found in the blood, and how many unique species we found for a given depth of sequencing. So it's not just disseminated pathogen. You would expect the opposite if it's just patients who have a bad infection and are bacteremic from it, should be dominated by staph or Pseudomonas. It's actually the opposite here. It was the longer the tail, the more unique bacterial taxa you found, the more acutely ill they were. And that correlation, that R squared of 0.16, if I did the same analysis for lactate here, it would be 0.08. And lactate's not a great biomarker, but it's what we've got for assessing is someone very sick. So actually this spray of what I think are gut bacteria, this may be an index of essentially gut wall integrity and whether it's failing or not. One luxury we have at the University of Michigan is the Weil Institute. We have a critical care research institute that lets us do things like a large animal lab that I couldn't possibly pay for with my own research expenses. We have a model of sepsis. We give the animals E. coli pyelonephritis, like the ureter, and clinically and immunologically, they get septic. They get distributive shock. They get quite inflamed. And it lets us do things like this. We can measure pulmonary artery blood, just going into the lungs, and systemic arterial blood, so carotid artery blood, and basically triangulate, say what's going into the lungs and what's coming out. And you can imagine if you want to, it's useful for what is the lung generating, right? The lung's a metabolic organ. If you see something being stepped up, it's being generated there. Or in this case, it's anything being filtered, something is stepped down. Quantifying the amount of bacterial DNA that we find. And these are all matched specimens from pre-lung and post-lung, pulmonary artery, carotid artery. And you can see that quite consistently, there's more bacterial DNA going into the lung than coming out, suggesting that the lungs are probably doing some kind of filtering role. Better at being, the lung has much more robust immune defenses than the brain, I have to assume, not a neuroimmunologist, but you'd rather there be filtering going on there before the systemic circulation. So I think that this data has collectively reignited the idea that gut leak and translocation may be an important pathway in multi-organ failure and critical illness, and something that we're not really thinking about as a treatable trait. Maybe we should be treating gut wall integrity as a patient phenotype that we should intervene on, or selective decontamination of the digestive tract, suppress the source community or survey for beyond. But that's not the only thing. Another way in which the gut microbiome is an important organ is its role in calibration of systemic and alveolar immunity. And I know that there are members of your department who won't believe anything if it's not in Nature, Cell, or Science. So here's something in Nature, Cell, or Science. Or if it isn't shown in mice. So this is all in mice. And there are remarkable studies that show the same thing. And that is, if you take our conventional mice, the mice that we work with in the lab all the time, the genetically identical Black 6 SPF mice, and you co-house them with mice from the wild. These are not wild type, but wild mice, right? Like from your basement, your barn, or the woods, right? The wild microbiome wins, right? So it takes over the artificial sort of bubble boy microbiome of our SPF mice, right? And these mice that have taken on a wild microbiome, and they've done really remarkable stuff with introducing them placentally, like it's from birth. It makes the mice healthier, they're more resilient to infections, and really concerning to me is they become a better model system for studying sepsis. So the young people in the audience may not know that back in the 90s, we cured sepsis with TNF-alpha inhibition, right? But you might notice that we don't give infliximab in the ICU. And it's because it didn't work. It was effective in mice. It was not effective in human clinical trials. It turns out if you run the exact same experiment with these wild microbiome mice that are now genetically identical to the ones that we did all experiments before, now it recapitulates the human response. It's no longer effective. So that's pretty concerning. A lot of what we've said is just different about mice and humans might actually not be true. It might be the fact that we've just completely distorted their microbiome, and all of the immune variation that we just think, the mature T cell immunity, is actually a downstream consequence of what we've done to muck up the microbiota. So that gives me pause and hopefully emphasizes that the immune response is important. There's another fraction of the audience that won't believe it until they show it in oncology. So here's 3 papers in oncology showing how important this is. And they all tell the same story. In patients with melanoma, we're trying to use checkpoint inhibitors for everybody, but they don't work for everybody. You have responders and non-responders, right? This is anti-PD-1 therapies. What they've done in the series of studies is, if you look at the gut microbiota of the people who do and don't respond, they differ. They're actually, in humans, doing fecal microbiota transplants from the responders to the non-responders, and some non-trivial chunk of them now convert. They become responders. Again, suggesting that the immune variation may actually be downstream of microbial variation. So again, just to get you enticed to think that, yeah, maybe there is something to the systemic immune consequences of the gut bacteria. And finally, what I'm beginning to think is the most important one is really the gut microbiome is a metabolic organ. So it is constantly taking carbon in from our diet, turning into small molecules that go into the systemic circulation and have profound consequences on end organs. So to show you how important this is, if we take our germ-free mice that have no microbiome and never had a microbiome and compare them with our conventional mice, there's more than 400 identifiable compounds in the blood of the conventional mice that aren't there in the germ-free mice. So it's really not just like you're missing the thyroid gland and thyroid hormone, you're really missing a bouquet of products that are gonna be, I'm gonna show you, influenced by our interventions like antibiotics. So 4 ways the gut microbiome functions like an organ. It prevents secondary infections, translocation of gut bacteria, calibrates systemic and end organ immunity, and dissemination of metabolites. So hopefully you're on board with, at least conceptually, there might be something to thinking of the gut microbiome as an organ. Moving on to the hidden driver of heterogeneity. So if you do critical care research, you better have the word heterogeneity in your grant proposal. It's definitely been the word of the last decade. We know that sepsis, ARDS, multi-organ failure, even pneumonia, I would say, in the ICU, are really not well-defined disease processes. What they are clusters of their syndromes, right? So the analogy would be it's 1850 and you have a patient with a fever. Some of them might be due to infection. Some of them might be due to malignancy. Some of them might be due to autoimmune disease. But if you don't have an understanding of the biologic underpinnings of that heterogeneity, you're going to treat them all with antipyretics and you're going to stop there, right? It's just going to be supportive care. And honestly, with sepsis, with ARDS, I think you'll all agree, that's basically where we are. It's basically still supportive care. And until we can get to the underlying biology that explains it, we're not going to make any more progress towards targeted therapies. So that's really the thrust of the field right now. How are we going to get there? You can take a representative, this is a prominent review on how we're going to disentangle clinical and biological heterogeneity in sepsis, published this year. And you'll see maps like this. You'll say, here's what we think is going on in sepsis. You have these features, the infection instigates it, and all of the molecular mechanisms are basically all of the omes you can think of. The genome, the epigenome, the transcriptome, metabolome, the proteome, and so on. And if we can just make some kind of sense of this chaos, the patients will sort themselves into clusters and maybe they might respond differentially to treatment. That's the idea. I've got major objections, these are my friends, major objections with this for a few reasons. I think the field made a major misstep in taking infection out of the molecular process of sepsis. I will talk your ear off about it sometime, but in work that we have ongoing, we found that you really cannot disentangle the identity of the pathogen from the host response. For instance, this is for the critical care nerds in the audience, the hyperinflammatory, hypoinflammatory subphenotypes from the Calfee group and others. What we find is that is overwhelmingly a difference in the identity of the pathogen. Hyperinflammatory is gram-negative sepsis, hypoinflammatory is all others. And I just think it's a real conceptual mistake to try to pretend like the bug isn't an essential part of the pathophysiology of sepsis. When I tell this to my ID friends, they're like, well, obviously, right? The bug is molecular pathogenesis of everything, right? But the other thing is there's no room for the microbiome in here. All those experiments I showed you where we can muck up the microbiota in our mice or our animals and change whether their organs fail or not and what their systemic inflammation is, there's no room for it in its conceptual model. So I'm going to try to convince you that we should broaden the margins a little bit and squeeze the microbiome in there. I'm also going to argue that if you want a biologic source of heterogeneity in your patients, the microbiome is where you should be looking. So any two patients in your ICU are 99.9% genetically identical, right? There's meaningful variation there, but by and large, there's a lot more similarity than there is dissimilarity. Let's look at their gut microbiota. So this is work with Bob Woods and Rishi Shandaraj, colleagues at Michigan. We looked at about 130 ICU patients. We characterized rectal swabs, and we see tremendous variation in bacterial density. So from 10 to the 4th, 10 to the 9th, just huge orders of magnitude of difference. And we have ways of showing it's not just due to variation in sampling. There really is this meaningful difference in just the density of bacteria in your lower gut that's modulated by antibiotics and predictive of clinical outcomes. And in terms of who's there, so this figure right here, every row and every column is a different patient. And where they intersect is how many bacterial taxa are overlapping. So 25 to 0. Some have very similar, have a lot of overlap in their bacterial composition, and some have none at all. So again, those two patients in the ICU, 99.9% genetically identical, may have no intersection at all in their gut microbiota. I think that that point is so fundamental that I've actually had ties made out of the figure and scarves. I'm not wearing it because I made the mistake of wearing it on rounds, and now I'm pretty sure that if I sequenced the tie, I would find the same bacteria that are actually depicted in it. So this has never been in the unit. So if you want to know where is the biologic heterogeneity in our patients, it is, I think, a colossal oversight to not include the microbiome as a major source for it. So what does this actually look like in an implementation? A lot of what we're doing is just starting from the ground up, asking if these core variations in gut microbiota explain clinical variation. That's one way to do it. But another thing that I think is useful is build off of the work that our colleagues using data science have already done. So people who have looked kind of agnostically at critically ill patients and said, what are the important and coherent ways that they cluster themselves? And then we can ask them, is that driven by differences in their microbiota? And I want to show that by embarrassing Matt Cherpek, who's in the front of the room over here. So when Matt was at another institution in the Midwest that I don't have to mention, he and Siva Bhavani did this very cool study. And I'd love to know why Matt got excited about temperature, because I'm now obsessed with temperature. But instead of doing what a lot of groups do, where you basically like feed a whole bunch of high dimensional data in and just say, do some clustering for me, what they did was they said, let's pick one variable, and that is temperature. And temperature, we know clinically is important. How many decisions do you make based on whether a patient's febrile or not, right? And they modeled it longitudinally, and they found that in every cohort they looked at, and so in a very robust clustering, I'm always a little self-conscious telling someone about their data in the room. So Matt, just throw something at me if I'm getting it wrong. But there are 4 distinct phenotypes. There are the people that come in hot, the hyperthermic patients, and are really slow to defervesce. The ones that come in hot and defervesce quickly, the normothermic ones, and then the hypothermic ones. And this probably fits your clinical intuition. You've seen the hypothermic patient, and it's usually like something's pretty wrong, right? Like they're probably frail and elderly. There's features of that. So it's very consistent clustering, and it seems to be really important. They're demographically different, they're prognostically different, and I think consistently so across cohorts, and immunologically different when you measure their cytokines. So there's something interesting. It would be surprising if there wasn't something interesting about that. Like I said, I don't know why Matt got excited about temperature, but I got excited about temperature because what does it tell us? It tells us about the patient's immune state and their metabolic state, right? That's why we measure temperature. Are they acutely inflamed, and what is their metabolism doing? And these are two things that I know the gut microbiome have something to do with. So we asked that question. We said, can we explain the Cherpek subphenotypes or the Bhavani subphenotypes? I don't know if they call them that, but the temperature subphenotypes. Is it possible that that's actually downstream of differences in their gut microbiota? So we started doing that, and this is all a project led by Cale Bongers, who's now at the University of Iowa. Again, Big Ten schools just cooperating with each other. And first, we started with Michigan medicine data. So in our ICU, we did the same kind of analysis that Matt did in his cohort and found the same 4 clusters. We had rectal swabs in about 130 of them, and we just asked, do they differ across subphenotypes, across cohorts? And they did, and what was surprising is how few bacteria you needed to explain the difference. It was actually a relatively small number of bugs that explained almost all the variation between them. And again, I won't make you memorize many bacterial names, but memorize this one, Lachnospiraceae. So it's a very common, abundant, lower gut bacterium. It's a gram-positive anaerobe. It's not classically thought of as a pathogen. And if you look at it, it explained most of the variation in the temperature subphenotypes, which is consistent across it. But this is, of course, all cross-sectional. This is just observational. Who knows? Maybe there was something different about the way these patients were being managed, differential antibiotic exposure. So we went to the lab and we modeled it in mice. So For this one, we were using the LPS model. So you take healthy mice and you give them endotoxin. And mice tend not to have fevers. They get hypothermic. They drop their temperature when you make them septic. And if you take a conventional mouse and you give it endotoxin, it drops its temperature and it recovers within a couple days. Germ-free mice, so mice that don't have a microbiome, have a much more blunted temperature response. They really barely drop their temperature at all. And look at the difference in variation. So we can take these 40 mice, and I should tell you, we got them from different vendors and different shipments. Why do we do that? It's so that the main source of variation is microbiologic. These were all black 6 genetically identical mice, same age, same gender, same exposure. We've basically controlled everything we can control, except we know because some come from Jackson in shipment 1, and some come from Charles River in shipment 2, they're gonna have different gut microbiota. Right? And you see huge variation in temperature. Some don't drop their temperature at all, some drop it profoundly. If we do the same modeling with germ-free mice, there's no variation at all. So really suggesting it's the difference in microbiota that explains the difference in temperature variation. And lo and behold, it's the same bugs explaining the temperature variation in our mice as it is in people. That Lactobacillus explains most of the variation. And to show you that the more relative abundance, the more lacno you had in the gut, so from 0 to 80% here, the lower the temperature loss in your model, right? So a third of the variation all explained by a single bacterial family in the lower gut. So I think this is exciting and interesting that some of this clinically important subphenotyping of temperature trajectories may be downstream of variation in microbiota. But what's also why I am still obsessed with temperature is because of this very weird study that came up in, a weird and important study that came up in dinner last night, which is, I don't know how many of you have heard this, but human body temperature has decreased consistently since the 1800s. I don't know how they got the idea to start studying this, but using a variety of data sets quite consistently, monotonically, 98.6 is no longer core body temperature. That's actually like the 1880s body temperature, and it's been steadily decreasing ever since. And no one knows why. They speculate maybe it's something to do with hygiene and latent infections. Everyone had smoldering fungal and mycobacterial infections back then, but look how consistent it is over time. What we know it's not is it's not changes in the human genome. Like that's not a meaningful time for human evolution to occur. It was happening before the onset of antibiotics. So what has changed surely since that time is the microbiome. So diet, hygiene, cumulative antibiotic exposure, the way we live, the way we interact with each other. So we've done a bit of work with healthy mice. This is in the same paper. For one, germ-free mice have a lower basal temperature than control mice, again, before you give them any exposure at all. No matter what enteric antibiotic regimen you give, you drop the temperature of mice. Again, this is outside of sepsis, no sepsis modeling here. This is just doing that. And it's not just because of food aversion and dehydration. This is a problem with you put antibiotics in the water, sometimes it tastes bad and they just dehydrate themselves. If you give it systemically, you can drop their temperature as well. So this is very concerning to my infectious disease colleagues because I'm giving animals antibiotics and their temperature's dropping, which we use clinically as evidence of clinical response, right? Oh, they defervesced when I gave them antibiotics. Here's healthy uninfected mice, and I'm dropping their temperature with antibiotics in the first place. And check this out, that same, I just showed you this, the more Lactobacillus in the gut, the more your temperature drops. We can take germ-free mice, monocolonize them with the most abundant Lactobacillus strain we've had, and drop its temperature, showing that it's not just an epiphenomenon, it really is causally related to the temperature drop. And it's a species-specific thing. It's not any bacterial species you put into the mouse. There are some that seem to modulate temperature more than others. So I have not moved on from temperature, but that's just emblematic of how what we think are clinically and interestingly important phenotypes might be downstream consequences. Of the possible explanations for why that could be, again, I think the gut microbiome as an endocrine organ is probably a very plausible mechanism by which temperature regulation happens. I have a grad student working on this now. As we've shown you, the more Lactobacillus you have, the more your temperature drops in your sepsis model. Mark is focusing on acetate. So as you've probably heard, gut bacteria make short-chain fatty acids. This is one of the most abundant bacterial products. And of those, butyrate is really important locally, but very little of it gets systemic. Acetate is the product that is most abundant both in the cecum and systemically. So we focused on that. And blood measurements of acetate track nicely with Lactobacillus in the gut and explain that same amount of variation in temperature. So again, same microbial variation in the mice results in acetate variation in the blood, correlates with temperature variation in our sepsis model. If we study acetate concentrations in the gut and the blood during sepsis, what we see is that it surges in the blood. So if I give a mouse LPS, we see acetate rise, and it plummets in the gut, which we haven't proven this yet, but I think a coherent interpretation would be that basically the gut is serving as a reservoir. kind of like your thyroid gland just stores up thyroid hormone, and then when it needs it, it dumps it. But acetate present in the gut permeability happens, acetate goes there. And does acetate matter systemically? Yes. We can augment the temperature response in sepsis by giving the mice acetate. So I'm not saying it's all due to acetate, but its most abundant bacterial product of Lactospiraceae does seem to recapitulate the physiologic effects we're seeing. And for Lynn's sake, in preclinical models of lung injury, acetate seems to be protective. So influenza and ischemic perfusion models, acetate does seem to have a role in terms of stabilizing epithelial cells in the lung. So that's going to be Mark's career probably, is trying to interrogate acetate. What determines its concentrations in the gut? What determines how much gets into the blood, and what its end organ consequences are? Finally, the most clinically relevant aspect of the talk, I'm going to talk about how the gut microbiome is potentially a therapeutic target that we should treat one day, but more accurately, it's one that we're already treating currently. You need to know that the healthy gut microbiome is by and large anaerobic. So 99+% of the bacteria in a healthy human gut are anaerobes. And anaerobic antibiotics disrupt the gut microbiome. So like your metronidazole, your clindamycin, like the ones that you're thinking of. But you probably also know that piptazo and cefepime, which you may have given once or twice in your career, we think of them as pretty equivalent. Patients come in with sepsis, give them vancomycin, give them vancomycin as relatively, but they're actually quite different when it comes to their effects on the microbiome. Piptazo kills anaerobes dead, and cefepime doesn't touch them. You would never give cefepime for someone who has an intra-abdominal infection, right? Piptazo gets into the gut in high concentrations, cefepime does not. And as a consequence, piptazo depletes gut microbiota, and cefepime doesn't touch them. This is not a subtle difference between them. So this is, again, data from Mark, a grad student. Here's a gut community from a bunch of mice who are untreated, no antibiotics, no sepsis, no antibiotics. And I can tell you that virtually all of the taxa here are non-pathogenic anaerobes. If we give the mice 3 days of cefepime, it looks about the same, really very little disruption. If we give them 3 days of piptazo instead, it looks like this. So the anaerobes are gone. You'll notice the y-axis is now up to 60%, Staphylococcus and Enterococcus, right? So they're really not equivalent when it comes to their effects on gut bacteria. One is essentially untouching them, not touching them at all, cefepime, and one is depleting them and making space for all these potential pathogens to overbloom. So I would personally rather have 2 to 3 pounds of this in my gut than 2 to 3 pounds of this. It also has a consequence on those gut metabolites. So I just tried to get you excited about acetate, so the most abundant short-chain fatty acid made by these anaerobic bacteria in the gut. This is how much we find in the gut of normal, healthy animals. This is how much you get after 3 days of cefepime. This is how much after 3 days of piptazo. So you've, again, induced a kind of organ failure. If you buy my argument that the gut microbiome is an endocrine organ, I have just completely made it devoid of the metabolite that we think is consequential. Most ICU patients don't need anti-anaerobic antibiotics, but receive them anyway. So there are pearls that we all got back in med school that are really hard to erase, but aspiration pneumonia is caused by anaerobes. That's not true. That hasn't been true for a very long time. Those studies lumped together patients with lung abscesses and empyemas. If you look at contemporary studies using culture or culture-independent-based techniques, they're really not the case. Urinary tract infections, you do not need, pyelonephritis, you do not need anaerobic coverage. It's really intra-abdominal processes, necrotizing soft tissue, deep head and neck. It's a relatively short list. There's this impressive point prevalence study done at the end of the last decade, 15,000 patients internationally, 8 countries. Only 0.3% of ICU patients had anaerobic pathogens, but half of them, 48%, got anti-anaerobic antibiotics. And I don't think it's because, I'm sure it was in some cases just sort of just in case reasoning. It's like, what's the downside? May as well treat for the anaerobes anyway, even if they're not there. But it's not really because they had a specific concern for an anaerobic process. It was more just what's the harm. And in multiple animal models of critical illness, lung injury, pneumonia, sepsis, I've shown you some of these. Pre-treatment with anti-anaerobiotics worsen outcomes. So if I knock down those protective anaerobes and give the mouse streptococcus, pseudomonas, or hyperoxia-induced lung injury, whatever model you care for, they tend to do worse if you take those, what we think are protective anaerobes out. Based on this, my lab and my trainee Rishi Chandraj and I looked at whether that difference in early antibiotic exposure among our ICU patients is clinically consequential. So did you or did you not get anaerobic coverage? Does that have an effect on your outcomes? So we started with thinking that this was going to be a story about VAP, ventilator-associated pneumonia, and we thought that the anti-anaerobe treated patients were going to have higher VAP rates. They did, but it was much more exciting than that. We looked at 3,000 mechanically ventilated patients at our center. Of those, 3,000 got antibiotics in the first 72 hours. You really can't be on a vent at the University of Michigan and not get antibiotics, right? But there was variation in whether you got anaerobic coverage or not. So 2/3 got pip/tazo or equivalent anti-anaerobic coverage, and the third did not. And there was a pretty dramatic, a 6-point absolute difference in 30-day mortality. And again, we expected it to be VAP, ventilator-associated pneumonia. That was actually a tiny part of this. It was really non-resolving organ failure. It was not getting off the vent. It was not getting off pressors. It was non-resolving kidney injury and so forth, right? So it's really more, I think, than just preventing secondary infections. So, but you might look at this and say, well, observational, descriptive, correlative, this is not really arguing for causality because there was something different about those patients. Someone decided to give you pip/tazo and you cefepime, so maybe there's some confounding by indication here. So we did our own animal modeling. Here's a Klebsiella, an infectious model of Klebsiella pneumonia. And if you pre-treat, again, this is not a therapeutic use of pip/tazo, this is pre-treatment. If you pre-treat the animals with pip/tazo, they got sicker, they had trouble clearing the bacteria, and they had higher mortality. In a sterile model, so this is oxygen-induced lung injury, I put conventional mice in pure oxygen, 95% oxygen. They will eventually die after 5 or so days, but in this case, on day 4, we turned a non-lethal exposure into a lethal exposure just by pre-treating them with pip/tazo, which just shakes me because I've given so much pip/tazo in my career and so much oxygen in my career. So here I am giving them together and showing that it increases mortality. So these are controlled animal studies, right? But you could say it's still just a single center. It's just the weirdos at Michigan who think everything is related to the microbiome. Some friends in the Netherlands. So this is Joost Bersinga and Bob Kohlberg. Looked at 16,000 emergency department patients, all comers, who either got anti-anaerobic coverage or not, did all of the same controlling that we did, and found a similar mortality delta at 30 days. But you could still say, same issue, observational data is confounding. How do you know that it really is causal? So that's when the heavens opened up and gave us the most amazing natural experiment I could ask for. So this is in partnership with Mike Shoding and again with Rishi Chandaraj. We leveraged the fact that some of you remember in 2015 and 2016, we had a pip/tazo shortage. We just couldn't get pip/tazo. And what I'm showing you here is the orange line is pip/tazo. It was about 1/4 of all antibiotic prescriptions in our hospital. On a dime, it dropped, and cefepime took its place. And then amazingly, when the shortage period ended, they had a complete reversal. So if you're a sort of clin epi nerd, this is just a gift from the heavens, right? This is a beautiful natural experiment where we're gonna have a lot of patients who got one treatment or the other based purely on calendar date, right? It was no other factor, and there's all these rigorous tests you can do to make sure it's performing well as an instrumental variable, and it does. And what's awesome is that we have before and after data. So if there were secular trends, you know, if people were just doing better in general because of different changes in practice, we can account for that too. So it's basically everything you could ask for from an instrumental variable, and it showed the same thing. So a 5% mortality advantage to getting cefepime compared to pip/tazo. For this cohort, this is really important, we excluded patients who didn't have equipoise. So if you've got intra-abdominal sepsis, if you've got a necrotizing, you know, like that's not kosher. I wouldn't randomize you in an RCT to cefepime to lack anaerobic coverage. So this is the population we're talking about here. So all of the observational data and all of the animal data is pointing in the direction of harm with anti-anaerobic therapy and benefit to anaerobe sparing therapy like cefepime. But those of you who are up in the literature will say, hang on, there was an RCT of this. So the guys at Vanderbilt did this really cool pragmatic RCT called ACORN. And this was among hospitalized patients. It wasn't sepsis. In fact, most of the patients didn't have sepsis. Most weren't in the ICU, but hospitalized patients who were gonna get either pip/tazo or cefepime. A little alert popped up and said your patient is eligible for a randomized trial. It's a very pragmatic EMR embedded trial. Will you let us randomize it? And they did. So half the patients got cefepime, half the patients got pip/tazo. Their primary endpoint was kidney injury, acute kidney injury in the first week or first 2 weeks. And it was a negative study, and they reported 28-day mortality and no difference. So you'd say, well, RCT wins, right? Importantly, they didn't care about anaerobic coverage. And in fact, their little pop-up said, if you're giving cefepime, consider giving metronidazole as well to cover the gap in anaerobic coverage. They didn't exclude patients who had a good reason for anaerobic coverage. And 50% of the cefepime arm got flagyl, got metronidazole, so a huge imbalance in terms of co-exposure. And because it was a pragmatic study, they couldn't stop the crossover, so 18% got crossed over. So good study, important for answering a direct nephrotoxicity question, but a very different question than ours, right? Like this was simply not comparing anaerobic coverage or not. And in fact, another grad student of mine is looking at this specifically on kidney injury. We redid their analysis using our data, right? So asking if effectively getting randomized to one of these drugs or the other changes your risk of kidney injury. And I think it was Hillary last night asked for this question, so she's in the audience, this is it. Here's patients who had sepsis, but didn't have kidney injury at onset, at arrival. And over time, if you got cefepime, this is the fraction, about 20% developed kidney injury in the first week. If you got piptazo, it looked like this. So higher rates of kidney injury in the patients who got piptazo. What if you got cefepime and metronidazole? You look like piptazo. So it really does suggest that it's the anaerobic coverage. If you deplete those gut anaerobes, you behave as if you got the piptazo. If I have convinced you, great. If I haven't, and you still think it's due to confounding, I would say, even if most of the effect size, so maybe you don't buy that it's a 5% mortality delta here. Maybe it's mostly that, but it's only a 1 to 2% meaningful effect, right? This is a paper done by Andy Abman, a colleague at Michigan, arguing that even that small delta, which may not impress the cardiologists in the room who are looking for bigger, or maybe not, that would actually be, given how commonly these drugs are given and what the baseline mortality rate is of these patients, actually really consequential at a population-based level, right? If you could improve the mortality of patients by 1 or 2%, given how commonly these decisions are made and what their baseline rate is, we're talking about potentially tens of thousands of lives saved. So I think that in sepsis, we've spent a lot of time debating time to antibiotics, getting in within 2 hours, getting within 1 hour. I don't think that's everything. I think which antibiotic may be more consequential. I actually think our data suggests if it takes you an hour to determine if the patient actually has an indication for anaerobic coverage, it's worth it, right? Like, because there's a real mortality trade-off here. The harms of antibiotics are not merely theoretical and distributed. That's commonly how we think of it, is like, I've got a patient crashing in front of me. I'm not gonna withhold antibiotics from them. The risk is potentially increased antimicrobial resistance across the population. That's everyone else's problem, right? I don't think that's true. I think the harms are likely concrete and borne by the patients who receive them. So I actually want to step up the urgency of stewardship discussions. Just in case antibiotic selection is really not a coherent strategy if our antibiotics have real associated harm. I mean, to make an analogy to cancer, the oncologists don't say, oh, let's just give them chemo just in case, right? Like, you have a good reason to do it because you know that these are very toxic agents, right? Like, and they have real harm, and you're gonna be very sparing and judicious in their use. And this is a little depressing, but de-escalation is not gonna save us. You know, in the ICU, we get away with murder. We can just throw the patient on Zosyn, Mirapenem, and everything, and Colistin, and then say, you know, in a couple days, we'll have cultures, and we can de-escalate and just put them on Unison and send them out, right? In terms of cumulative antibiotic exposure, antimicrobial resistance, maybe that's a good thing. In terms of the microbiome, it won't help. I think, honestly, a dose or two of piptazo and those anaerobes are gone, and it's probably weeks or months before they come back. So, and we don't have a good way of giving them back in the ICU right now. So I really do think that we have to force the issue at the upfront decision-making. This is really an ED, urgent care, transfer from the floor to the unit decision. It's really not something you can punt until the next day. To wrap up, I'll say the hardest way to fix the microbiome is by engineering an ecosystem. I've been very disappointed and unsurprised by how most of the probiotic studies in the ICU have been negative. If you're even aware of them, here's a big one that was in JAMA a few years ago. They gave lactobacillus, an oral probiotic, to patients hoping it would prevent VAP. It had no effect on VAP. More than 80%, if you look in the supplement, more than 80% of the patients in this study were getting active antibiotics that kill lactobacillus, right? So this is like, you know, there's an active forest fire, that's the Zosyn, and you're trying to plant seeds while it's happening. That's not gonna happen. I just think it's very unrealistic. And it's not, these are immunosuppressed patients, and a lot of them actually did end up developing seral site infections with the bug. So I don't think willy-nilly we should just be giving live bacteria to our septic patients. I think we need to be more thoughtful than that. What's hard, but maybe more feasible, is actually repleting the microbial products. So don't give the bacteria, but give their bacterial products. And this is something we're working on with our pig model, is blasting them with piptazo. And then this is my partner, Kathleen Stringer, whipping up a cocktail of butyrate, propionate, and acetate, the short-chain fatty acids the bugs make, giving it by way of enema, and showing that it can actually restore blood concentrations and hopefully have restorative effects on the end organs. So I made the joke last night, for Kathleen's sake, I kind of hope this doesn't work, because if it does, every ICU nurse in the country will curse her name, because any doc ordering piptazo you have to give the smelly butyrate enema to make up for our injudicious use. And then the easiest thing should be minimizing dysbiosis in the first place. Like, really, I'm not asking much. I'm saying only give anti-anaerobic antibiotics if there's a good reason to. No one's ever shown superiority in all-comers community sepsis, so there's no affirmative clinical evidence. The best you have is equipoise, and all the observational, all the experimental data point in the direction of harm. So I think it's an interesting discussion of whether there would be equipoise for a prospective study now, like would I really let my level 1 be randomized to Pip/Tazo if there's not an indication for anaerobic coverage, but that's a debate to have. And then I'll leave you with a futuristic idea, which is can we actually engineer gut bacteria to perform specific functions? So this is a really weird but really cool and provocative study from a few years ago in Nature Medicine. Don't ask me why they were doing this, but they were studying gut microbiota in marathon runners. In the gut microbiota of people upon finishing marathons, you see a surge in a specific bacterial species, Veillonella atypica. And they knew about this bug that it only metabolizes lactate. It's only got a single potential carbon source, and that's lactate. Marathon runners generate lactate from their muscle and into the blood, and they said, is it possible that the lactate's getting into the blood, going into the gut lumen, getting metabolized into these short-chain fatty acids like propionate, getting back into the blood and improving performance? And they did a whole series of experiments in mice, yes, marathon mice, with microbiota transplants from these marathon runners, and it worked. And that's really kind of mind-blowing, right? Like when we say lactate is confounded by what? Kidney failure and liver injury, it's metabolized. It might also be confounded by gut microbiome failure, right? Like it's a third source of metabolism for it. And more important than getting rid of that lactate, we may be depriving ourselves of the upcycling, the beneficial products that it generates. So we've done some preliminary work, this is all unpublished, using our instrumental variables. So using that Pip/Tazo shortage and asking if you happen to get Pip/Tazo, anaerobe depleting, in pink here, versus cefepime, is there a difference in your baseline lactate? So these are all septic patients, and whatever your, it's supposed to be a measure of perfusion and organ function, right? Whatever your MAP, whatever your mean arterial pressure, lactate was higher if you got Pip/Tazo, if you got the anaerobe depleting ones, right? And your trajectory was slower. So lactate clearance was indeed impaired if you got the microbiome depleting therapy. And I'm working with Jiahe Li, who's a biomedical engineer at Michigan, who is knocking lactate oxidase. So this is an enzyme that metabolizes lactate into a probiotic, giving it to mice, and we've shown that it can actually decrease baseline lactate levels, and in 2 models of lactic acidosis, so just giving them lactate or boxing their mitochondria with metformin, we can impair lactate clearance or accelerate lactate clearance by training the gut microbiome to metabolize lactate, so treating it as a lactate sink. So that's what, in terms of what we may be doing coherently one day, is targeted use of this end organ, the gut microbiome, as a metabolic resource for solving our problems for us. So with that, done with that, and I want to thank my lab, my collaborators, and again, thank you all for the invitation to speak. Really happy to be here.
Great, thank you. Really intriguing. I'm going to ask the first question. So one of the other variables in the ICU and that impacts the gut is our feeding
Absolutely.
protocols. So how does being NPO or trickle feeds or full interval feeds fit into this whole paradigm?
Excellent, excellent question. And I have been in the unsavory position of giving a talk to Aspen is our nutritional conference, right? This is like all the people who their lives are on ICU nutrition. And I basically had this message was nothing that you do matters, at least with regard to the microbiome. Let me finish. So long as we're blasting our patients with Pip/Tazo, right? All of the fermenters, right? Like all of what we think are the key metabolic pathways that we want the gut microbiome to do, we've just eliminated with our anaerobe depleting antibiotics. I absolutely think diet, whether you're feeding, what you're feeding them with, most of our tube feeds in the ICU are not microbiome oriented. They're simple, they're rapidly absorbed. We're not giving them starches that the gut microbiota can do it. So I'm not saying to do it, but I'm just saying like, even if we are doing optimized feeds, I don't think it's not a microbiome therapy there. I think that things like feeding, things like PPIs, things like oral hygiene, all these things surely have consequences in the gut microbiome, but the effect size is this compared to Pip/Tazo or cefepime. You saw in my mice how dramatic that difference is. So I think variation in antibiotics is the low-hanging fruit now. I think that's going to overwhelm what are subtle differences. That's not to say there aren't other non-microbiome pathways by which nutrition is important, but in terms of microbiome pathways, I think we got to fix the gut microbiota before we think about how to feed it.
Question for you. So I'm Mark Monajet, infectious disease. So the new fecal transplant we use for C. diff is essentially spores. Is there any studies I'm not aware of, or I haven't come across it, that would change outcome without C. diff to give it to patients who are
Excellent.
critically ill?
If I understand the question, it was fecal microbiota transplant, which is, it's a shining example of a microbiome-directed therapy that's worked in refractory C. diff. I will say it's been somewhat disappointing for other indications outside of the ICU. It doesn't fix obesity, it doesn't fix metabolic syndrome. Like there's things that we hoped it would do. In the ICU, it is, as you probably know, it is sometimes being used for severe refractory C. diff. So whereas those patients might have gone to the OR for a colectomy in the past, we're doing it. And it's, I would say at the anecdote and case series level right now, I'm not aware of robust studies doing it. And I'm certainly not aware of anyone doing it for a non-C. diff indication in the ICU. Because
even in the outpatient setting, we have people who are having like 5-year cure instead of C. diff, and we give them the commercial product Vost, and they are like better in a week, and it's gone.
Yeah, I'm not opposed to it. I only had a slide, I think, on the probiotic studies. Even our dinky little probiotics can still sometimes have unintended infectious consequences. So it just makes me nervous. Like these are really vulnerable immunocompromised patients in the unit. And to me, fecal microbiota transplant is not the direction we want to be headed. In the era of precision medicine, it couldn't be less precise, right? Like you're just giving them someone else's microbiota. It just feels to me like it's fraught with, we need to be a bit more, you get what I'm saying, I think specific therapies are really where we should be headed. And I'm not gonna be the one leading the charge for FMD trials in the ICU.
Although I was going to go for the elite athlete microbiome.
Sure, yeah. You can pick your.
So there's some questions online I want to get to. Okay, so from Dr. Barzi, how does aging influence your mice and human data related to lactose? How do you pronounce it?
Lactose brachiae, yeah. Or you can say lactano
Populations and attenuation of temperature changes as
Was
well.
that aging?
Yes,
What's the question
aging.
there? I love that question, and it's not something we've looked at. We haven't done this on aged mice, but you're absolutely right. These things feel related to me. The hypothermic patients are disproportionately elderly, right? And I think we see that clinically. And I don't know if there's already a good host side explanation for that, but I can tell you a few things happen when you get elderly. Temperature drops when you're sick. Sepsis rates go like this. You know, sepsis is really U-shaped. It's the very young and the older. And your microbiota do change. I have submitted a grant proposal asking if we can tie those things together, and that one didn't hit. But I'd love to know is it possible that some of those metabolic and disease susceptibility differences in the elderly are actually downstream of differences in gut microbiota? And maybe it is cumulative antibiotic exposure or changes in diet. Or it could also be an artifact. It could just be that elderly patients happen to have different gut microbiota. So I love the question, but we haven't done the aged mouse experiments yet.
Back to your very original selective decontamination. After everything you presented, the last study I think I would do is a selective decontamination study, because what you said is more bacteria, breadth of bacteria, species. Explain, why did selective decontamination work?
I love that question. And in terms of the implementation science question of why hasn't it taken off? I think it's because people don't really understand it. The S is for selective. It's not broad spectrum. It sounds like it. You say like it's 3 drugs, right? But they quite specifically are just suppressing gram negatives, the aerobic gram negatives. So it's not that all bugs in the gut are protective and advantageous. The ones that it's suppressing are the ones that we're familiar with. It's the enteric gram-negative rods that cause a lot of trouble. It's the Klebsiellas and the E. coli's and the Proteus and so forth. It quite deliberately is sparing of anaerobes. And if you talk to the Dutch, they would say SDD, selective decontamination, is not just a drug. It's not just an intervention. It's a whole sort of suite of practices. They'll do things like ecologic rounds in the unit. So they'll say, we've had 2 gram-negative rod infections in the south side of the unit. Maybe we should up the SDD on that side, right? They do daily or every other day gastric aspirates to see what's growing in the upper aerodigestive tract. And importantly, part of SDD for the Dutch is avoiding unnecessary anti-anaerobic antibiotics. And they would say, if you're doing an SDD trial, the most recent big multi-center one was Sudiku. It didn't have any constraints on antibiotic prescription. And I would say, does SDD make sense if you're getting Pip/Tazo or Unison? And said, no, not at all. You get what I'm saying? Is like, what are you trying to save anymore, right? Like you're trying to selectively spare the bugs that you've just depleted. So it really is two sides of the same coin, but your question, I think, reflects a very common misperception, which is that it's just empiric prophylactic, just kill all the bugs, which I think is not actually how it works.
I'm going to paraphrase a question from online. So Wisconsin, I would assume Michigan has a large rural population, as well as urban areas. What is the difference in the microbiome of a rural farmer versus urban, and does that impact susceptibility to disease?
I love this because it brings me back to the lungs. The best data on that comes in children being raised on farms, have lower rates of A to P, asthma, secondary. And I think there's Erica von Moodius in Germany has done extraordinary work on this. It's a pretty tight story about upper airway microbiota, cumulative exposure to farm-related microbiota, mediating the protective effect of farm living. And it really is an intensive farm living there. I'm not aware of anything on gut bacteria, and certainly in ICU patients. I can tell you we're part of the APS consortium, so ARDS pneumonia sepsis is a big NIH-funded multi-center. It's going to be 4,000 patients observational study to try to understand this heterogeneity. NIH cares very much about understanding differences in rural and urban outcomes and what the explanations for that were. Is it all just time to transfer and acuity of care and things like that? I think that might be an opportunity to ask about it. I just hope that they don't all get Pip/Tazo at the pre-treating hospital by the time they get there, because the effects are quick. We see it within a day. So great question, but I can't speak to it specifically for this disease process.
Okay, we were just at time, so I want to thank Bob again for an amazing talk.